This afternoon, we are interviewing people to see their knowledge on the effects of drugs and teach them new information. We have recently studied the effects of different drug concentrations on Planarian motility. We put planaria in 1 millimeter concentrations of sucrose, nicotine, alcohol, and caffeine for 2 minutes and counted how many lines they crossed on graph paper. We repeated that for 3 more trials. Next, we put planaria in 10 millimeter concentrations of sucrose, nicotine, alcohol, and caffeine for 2 minutes and counted how many lines they crossed on graph paper. We also repeated those trials for 3 times. We also recorded how many lines they crossed on graph paper in water. The purpose of our experimentation was to see how planarians responded to different drugs and different concentrations of drugs. Planarians have similar nervous systems to humans, so by testing drugs on planarians, we get an idea of how humans would react to the drug. For this experiment, there were many different materials involved. This includes petri dishes, sucrose, nicotine, alcohol, caffeine, and the control, water. It also includes planaria, a timer, a camera, and graphing paper. A rehab dish is also needed to place the planarian in after they had been tested on. The first drug we tested on was nicotine, which travels to the brain quickly and generally creates a feeling of a racing heart. We interviewed a teenager off the street. Let's see how much they know about nicotine. Today we are with Helen, age 14. Helen, how do you think nicotine affects your body?
I think that nicotine makes you really calm.
How do you think the higher concentration of nicotine affects your body?
I mean, as the concentrations get higher, you would get calmer.
Helen was wrong.
In the higher concentrations of nicotine, it was found that there was more movement because nicotine is a stimulant. This graph clearly shows the effect of the stimulant.
There was a large range between concentrations. The second drug tested is alcohol, which is a depressant. We asked another student off the street her opinions and beliefs on alcohol, and let's see how correct she really is.
Hi, my name is Charlotte and I'm 15.
How do you think that alcohol affects the human body?
I think that it affects the nervous system and makes you more hyper.
How do you think that the higher concentration of alcohol affects your body?
I think that it makes the conditions more intense.
Thanks, Charlotte. Charlotte was also wrong.
With alcohol, it was found that the planarian movement decreased because alcohol acts as a depressant. In this graph, it's clear to see how the depressant worked on the planaria.
Alcohol had the largest range. Next, sucrose was tested. Sucrose is a carbohydrate which produces energy when consumed. Let's see how much the next person knows about how sucrose can affect the body.
Hey, my name's Nate, I'm 25.
Okay, Nate, how do you think that sucrose affects the human body?
Like you put on weight, man.
How do you think that higher concentrations of sucrose affect the human body?
You get like really, really fat.
Do you think that it affects your energy in any way?
Yeah, you get more energy.
Okay, thanks, Nate. Nate was right because sucrose does give you more energy.
Sucrose acted as a stimulant, so the planarian movement increased.
The range between concentrations was not as great as seen in the other 2 drugs, but was still great enough to prove that it is a stimulant. The last drug used was caffeine. Caffeine is a central nervous system stimulant. Let's see how much people know about how caffeine really works in the human body.
Hi, my name is Brittany. I'm 32.
How do you think that caffeine affects the body?
I think that it makes you really jittery and hyper.
How do you think that the higher concentration of caffeine will affect your body?
I think that it will make you very unhealthy and very hyperactive.
Brittany was correct.
Caffeine is also a stimulant and increased the planarian motility.
Caffeine overall had the highest amounts of planarian motility.
Caffeine had the lowest range between concentrations.
Finally, planaria were placed in water as a control to see how the different drugs affected the planaria. We used this group as a baseline to compare the other results to. Generally, the planaria moved about 8.75 lines in 2 minutes. Now it is clear to see how the different drugs acted as a stimulant or depressant. Drug addiction has many different psychological and physiological effects on the human body. Many people do not realize the harmful effects of what they intake daily. Those individuals who are unaware of these toxic side effects will benefit from the discoveries of this study.
Our hypothesis was if caffeine at the highest concentration is being used, then the planarian will have the highest rate of motility. Our hypothesis was correct.
There were no major errors in this experiment, but one improvement that could be made is to repeat the trials to reduce human error. Further experimentation ideas is to test different concentrations of each drug to see how they affect the planarian motility, and to also test different drugs to see if they have a different effect on the planaria.
During the acute stereotypy lab, we learned that stereotypy is the repeated movement, and in planaria they move in C shapes, which replicates a seizure.
In the repeated stereotypy lab, we learned that when planaria are exposed to drugs many times, that they build up tolerance and develop dependence.
In the acute stereotypy and drug interactions experiment, we learned about synergism, potentiation, and antagonism. That experiment showed how different drugs combined could have different effects overall, and it could actually reduce the effect, have no extra effect, or boost the effect of the drugs. We believe that all students should participate in this experiment so that they can learn about the effects that drugs have.
We also believe that they should learn about the body and learn about which parts get affected by the drugs.